In this video, I'm going to demonstrate the quality assurance and analysis features in the current version of the ArchiveFlow prototype app. I'm using simulated data here, but the place in the workflow that this represents is after the user has successfully created a local directory to store the primary data, and that primary data has been extracted. The user needs to select the cohort and the corresponding subject session in order to view the data. The first thing that's going to be extracted and presented to the user is the names of the streams associated with stream types. So TTL is associated with wave 1, the isobestic stream is 405A, and the experimental stream, which is the calcium signaling channel, is 407D. The sampling rate for each of these is shown, and it's going to confirm that in the TDT binary directory, these are considered streams. The next thing the user can manipulate is the lowest smoothing fraction. This controls how smooth or how wiggly the curve is after the lowest smoothing. Lastly, next they can control the downsampling rate. These were recorded at around 1,000 kilohertz, or sorry, 1,000 hertz, and you can downsample to 10. That's the default sampling rate for most of the analyses we deal with. You have the option of downsampling and smoothing and downsampling, which is computationally more expensive but gives typically a better result, and then downsampled only. So by default, we do downsampled and smooth because a lot of the computation that is done on older machines can't handle the smooth and downsampling. But we'll switch this now. So you'll see that it's going to take a little bit longer. The other thing that we can control is the filtering parameters. So you can filter by TTL. TTL is a binary stream that indicates when an attached recording device is actually recording. So for all the analyses that we've been working on, we typically want to filter by TTL. Lastly, you could control the offset if you wanted to manually shift the start time for whatever reason. So the first type of plot you're going to encounter is a signal QA plot, and that is basically to allow you to force you to physically view the smoothed, or in this case, smoothed and then downsampled signal. So the nice thing about these are plotly plots, so they're fully interactive. So you can zoom, you can pan, you can even download plots as PNG for that particular viewpoint. So it allows the user to get very close with the data. So here you can see that this simulated data is simply an exponential bleaching curve with an artifact present. We call these box artifacts. That box artifact is present in both the experimental and the isobestic signal. And clearly the experimental, there are very clearly simulated calcium responses here. And again, you can zoom in as you wish. So that is what it looks like for simulated data when you do the more computationally intensive smooth and downsample. If we switch to downsample then smooth, you'll see that it is quite a bit more jagged. And again, that is just simulated Gaussian noise. All of this is simulated data. So we present the user with the ability to label these box artifacts. We show them a photo bleaching corrected curve, so the box artifacts are more prominent. So this box artifact occurs roughly between 4,800 and we'll say 5,700. So the user is allowed to manually label these. So that will be saved for this analysis in all of the metadata that this app is going to generate. And next you can do the same for the experimental channel. So again, we can go back to isobestic. So that was 48 to 57. And so this will be maintained through the entirety of this. And then when you have confirmed that you've looked at this data and you are happy with it, I'll go back to a smooth and downsample so you can see how smooth this is. You can confirm for experimental, confirm that you looked at the isobestic signal, and then you can go to the second kind of plot, which is the isobestic and experimental plotted together, and then the delta F over F. This is in the normalized, and then this is z-score. And again, if you switch to smooth and downsample, you will see a much clearer response. Confirm these. Once it's sensed that you have confirmed the isobestic QA, the experimental QA, and then you've looked at the smoothing, which is the delta F over F, you can save the subject data and you can move on to the next subject, of which I have not simulated. So we'll add the behavior information. So rather than force the user to go through a consistent naming scheme, the alternative to this is to essentially select the session and the subject and then allow the user to upload the corresponding behavior data. This means that you don't have to keep things as neatly organized, and so it's automated. You can essentially upload these to the app. That's one of the benefits of Streamlit. So we'll browse these files, and we have the simulated baseline behavior results. And we will write the behavior data, and we'll continue on to the next subject, which doesn't exist. So we're going to show off some of the things that the behavior analysis can do for the simulated data. You'll select the mouse, and then you can control how much time is padded before and after the tone event, and you set the tone event duration. The way the experiment is designed is that there are tones that are presented, so you need to essentially extract and time align the delta F over F based off of the tones. So we have two types. We have CS plus tone and a separate CS minus tone. This is all simulated, so I'm not going to get into what that means. But essentially, you can view this data as it is aligned. So this is, again, the delta F over F. And you can save all of this aligned data. It's going to generate the data in the simulated mouse. And this is going to produce a CSV of both the of these curves that you can use for statistical analysis in software applications like Prism. There are additional analyses that can be done, but the simulated data does not contain a simulated mouse trace or position or anything like that. So the key point I wanted to make here with this demonstration was that you can add in analyses. It'll save the parameters in which you selected, and it will allow you to create reproducible analyses. So in order to prove that it has paid attention to the parameters you selected, we'll go to the subject status page. This overall table will allow you to see for individual subjects and sessions what has been done. So we have created streams, we've processed the streams, as I showed you before, and we created an associated behavior. And then if you want to look at the streams table, it will show the tank order. If TDD tanks contain two subjects, this is the first subject in the tank and the only one. It'll show you the TTL stream, the ISO stream, what was selected, so you can quickly glance at this table and see what was done. For processing, these are the, some of the parameters for the exponential decay function that represents photo bleaching. In smoothing, you can see that you've selected the smoothing fraction and the sampling rate, and you can sampling rate, and you can see it's TTL filtered. And for QA, you can see the signal ratio and how many isobestic artifacts and experimental artifacts were selected. In addition, you can see for the behavior data, it will say that you used this ground truth type. That's baseline. So in addition to what I showed you behind the scenes in the application, you can upload a template for what the behavior is supposed to look like. And if it does not match that template, it will send an error. And it will tell you where the original behavior file came from. We didn't fit the regression model because it didn't simulate the data, so we'll skip that. And then lastly, you are allowed to kind of explore the data. This is the normalized delta F over F. And as you can see, it saved the somewhat noisier downsampled and then smoothed. But you can choose which tones you want to see. So that's the CS plus, first, second, the third. You can show box artifacts. I don't believe we have speed, so that's not going to work. But if you had speed associated, you could see that plotted over. And you can also create custom subsets. So if you were interested in particular time points, let's say between 200 and 300 for whatever reason, you basically say, I'm interested in time point 250. That's where it will start. You select how much time you want to view pre and post. We'll say 50. And it's going to last for about 50 seconds. And you can see that it's going to present that individual trace of what you selected. And you can download this actual raw data into an Excel workbook that you can use whatever additional analysis software you want. So this is a brief overview with simulated data of what you can do for analysis and QA with the current prototype version of ArchiveFlow.